Good afternoon. This is Dick Brostrom from CDC on the COVID response here for the Pacific Islands. We're pleased to bring to the local jurisdictions the lecture training course number 7, the 13th and 14th lectures of this series. You can see we've marched through this these sessions in the weeks prior. A reminder that all of these sessions are recorded and the lectures themselves are available for download in PowerPoint format for you to share with your clinical staff, nursing or clinician or physician. And we anticipate that will also be true for today as well. I think all 6 are loaded up already on that PHOA website. They've been very kind to support this for us. Today, as you can see, we want to talk about interesting and important lab updates, so the clinicians are knowledgeable about the lab tests that they will be ordering or maybe have already been ordering. And then a discussion that I'm really looking forward to as well for what we know and don't know yet about the vaccine. As usual, I'm going to begin with a quick look at the USAPI and our status. I know our clinicians are not privy sometimes to the public health announcements, although you're very, very much aware of what's going on in your communities. The only 2 jurisdictions here with cases in the USAPI, and I remind you this is a log scale. So the pink line there, which is CNMI, it has about 42 cases, I believe. The rest are in orange are Guam. I like the flatness of that curve. That curve was pretty steep in the beginning. It's flattening out, which is a testament to the amazing effort by the public health programs that are there. Here you can see with those 2 jurisdictions that do have cases, lots of testing going on. Almost 40,000 tests have been done already. And then proudly 4 jurisdictions that are still some of the few locations in the world that are COVID free. That doesn't mean they're free from COVID worry, however, does it? Something to share, as much as these islands, many of them are feeling intense pressure, mostly politically, but also economically, to open up for repatriation of efforts for students, for patients who were stranded on medical referral in Hawaii or Philippines or in Guam, for family members who want to return for celebrations or for funerals. So there's lots of pressure for the local jurisdictions to find a way to safely bring mainlanders into the jurisdictions. And just wanted to remind you, we do have some data that was provided to me from Jess Tudela and the CNMI folks. They have screened 2,000 people. And overall, what they found is almost 1% of the people they've screened, even though many of them were screened prior to departure, almost 1% are unknown new test positives upon arrival. Some of them are diagnosed at the airport. Some are diagnosed during the 5 days of quarantine that's required in CNMI. When you look at returning residents, that number is a little bit lower, which is reassuring. They have not let tourists in yet, so that number, we'll know more about that going forward, because I think there's going to create a bubble with Korea and there should be some flights coming in. Among returning workers who are non-residents, however, the rate's pretty high. You know, we like in public health to look at number needed to screen to find a positive test. So overall, for every 126 passengers, 1 is going to be positive. If it's a returning resident, only about 1 in every 200, but that still means 1 out of every 200 is COVID positive. For tourists, again, we don't have data yet, but for returning workers, 1 out of every 60 that comes back to the island is testing positive. Now, a big credit to CNMI is that they know this is happening. They're very much aware, and they've been able to go almost 50 days, I believe, without a test being positive in their community. so that they're finding these travelers in their quarantine or at the airport. So it hasn't yet resulted in additional community spread. So again, COVID is trying hard to get onto the COVID-free islands. So open with care, as we say. You guys have created an amazing bubble that most of the world is jealous about. But we do know that we don't know how long politically or economically that can last. Reminder about the treatment guidelines that were put out last week. They're excellent, they're concise. It's a quick read, basically in short, remdesivir, which most of you have access to a little bit of it, not a lot. It can be prioritized for patients who need supplemental oxygen, but not on the ventilator. And dexamethasone, which we have a lot of, is mainly for the folks who are needing oxygen and or are vented, including those who are pregnant. Additionally, we still realize that there's a less experience on a ventilator with mass casualty events or mass population events than most folks are comfortable with in the region. We talked about ventilators in our first lecture, including how to hook them up and get them running. We've shipped, the ventilators were shipped by FEMA. PHOA has helped to ship regulators and high pressure oxygen hosing so that we can connect them to the large green tanks for those hospitals that don't have centralized oxygen delivery. In addition, with the great cooperation of actually the embassy in FSM, the Spirit of America is going to provide additional ventilator training. I think that this would be particularly possible for RMI that may request training like this, also in Palau. Right now, there's 16 folks who have been allowed to sign up for this in FSM, but it could be made available to the other jurisdictions upon request. And there's a course registration online. There's no cost, but there is like about 10 hours of online coursework that each clinician would have to take first, and then you would be able to avail yourself of live training with the team and a certified instructor from George Washington University, and then a possibility, depending on travel restrictions, for on-site training, all of it free of cost to the local jurisdiction. So if you're concerned more about ventilator management, this will take you from the basics to some fairly advanced training. Okay, our first lecture is going to be about testing, lab testing. Reminder, this is the lab testing you have here. This slide's actually out of date. I know that Majuro is quickly coming up to speed with an RT-PCR. American Samoa is getting ready to start testing. I think they actually have done some testing, their first round. Palau has been doing testing with the RT-PCR. So has Guam and CNMI. Other jurisdictions getting closer. I know so much more about this than I did in February because we really need to learn about this for managing our cases. And we've been able to assist with shipping both the Gene Expert test, and all of you have this platform, and all of you have used it in your jurisdictions, and the Abbott ID Now test, and we'll have some discussion of the sensitivity and effectiveness of these tests. They're both very good tests, okay? At one point, Abbott was considered the Cadillac for the whole world in terms of point of care. Gene Expert is probably a little bit better, but they're both great tests. A total of almost 100,000 tests have been, or soon will be, made available to the jurisdictions. I can tell you any state in the US would love to be sitting with 20%, enough testing for 20% of their population and have it ready to go. So your labs have it ready to go, but you have to order it. Some jurisdictions have begun this week to order the way they should for PUIs. You know, you might find your first case in the region by someone who is in quarantine. That's probably the most likely place you'll find it, is a repatriation in quarantine. But your first case might come from someone standing in front of you in clinic with fever, cough, or any of the symptoms you see here. So order these point of care tests. Your lab stands ready. Just a couple of jurisdictions still have yet to be successful in ordering and running this test. Of course, the nasal swab is no fun for your patient. And some jurisdictions are still trying to figure out who has to do the swab. Is it the doctor? Is it the nurse? Is it the lab tech? So get that figured out and get these tests ordered, and then we can all feel more comfortable that this preparedness effort can be set aside as we begin to turn our attention towards other important things. With that, I want to introduce Dr. Owen Chan. Dr. Chan is an assistant professor in pathology at the John Burns School of Medicine here in Honolulu. He's respected in our community greatly. He's also the director of the Pathology Core Shared Resource for the University of Hawaii Cancer Center. And so it's been great to be communicating with him over the past couple of days as he's prepared this talk for us. So Dr. Chan, I'd like to stop sharing and let you take the screen. We're close. Also, make sure you're not on mute. Dr. Chan, if you see over where it says display settings, you'll want to click on that and go to a single, yeah, there you go, duplicate. No, the other duplicate slideshow. Yeah, it should work. There you go. Perfect. And we're still not hearing you, sir. Stand by, folks. We're getting close. Dr. Jen, you're having some issues, I think, with, we're seeing your slides very well, but we are not hearing you. Let's take a look and see if you're on mute. You are not. Okay, Dr. Chan, I think you've sent me the slides, so I'm happy to push them for you. So the main issue here would be your ability to talk to us.
She may want to restart her computer. Sometimes Zoom will glitch, and then if you restart your computer, it's fine.
Yeah, Dr. She's also, there's also been a request to have the second speaker go first.
I see that. I'm not sure that he has, if Axel is online, let me check. Yeah, Axel is online. Axel, are you able to go first?
Oh, okay. Well, I mean, if you prefer me to do that, then let me just quickly get my presentation up.
Wait, Axel, I got an introductory, a quick introduction for you. Hold on 1 second.
Yeah.
No worries. Stand by, folks. Okay, can you see my screen?
Yes.
I can.
Okay, great. So before we hear our vaccine lecture, I have a few questions for Dr. Axel, who's Lair, who's going to be giving us this next talk. Number one, please tell us when the vaccine will arrive. Will the U.S. Pacific Islands be prioritized or left behind? Who's considered the highest priority for vaccine? How effective is the new vaccine? How safe is it? Will it actually prevent COVID-19 infection, or will it mostly just limit the severity of illness? And knowing most COVID infections are not diagnosed, what happens when the vaccine is given to someone who's already had COVID-19? Will the vaccine be appropriate for all ages? And how many doses will we need to be protected? So, of course, there's a little bit of snarkiness there because I know that none of those questions can actually be answered at this point. We're just not to that point yet. So that's not to disappoint anybody, but just to prepare us that the vaccine is a work in progress. It's an amazing, phenomenal, unprecedented work in progress. But a message to the clinicians, I really need you folks to be pushing into your public health programs to get them, help them get ready. In general, as clinicians, we'd like to let public health take care of this for us. We'll see the patients, let them get ready for us. But I want you, especially those of you that have any public health experience, to get involved, to make sure that your jurisdiction is ready. Do you have a vaccine plan? For those that have outlying islands, does it include petrol and boats and personnel that you're going to need to do the job? Is your cold chain ready? Do you have enough freezers and refrigerators? The time to order this stuff and be prepared is probably now, because when the vaccine is ready, October, November, December, January, who knows, there's going to be a major global push to procure this. And just like trying to get gene experts, if we're late in getting things ordered, we could be left behind. So I think we have to make some bandwidth here. It's hard to do because we're so busy with everything else, but leaning forward, please help your jurisdictions to be ready for the final solution, which should be vaccination for COVID. And so with that admonishment and hope, I'm going to introduce Dr. Axel Lair, Assistant Professor of the Department of Tropical Medicine and Medical Microbiology and Pharmacology at the John Burns School of Medicine. Dr. Lair, we look forward to hearing what you have to say about the COVID-19 vaccine. Go ahead.
All right, thanks for the kind introduction, Dr. Brostom. I'll try to share my screen now. Hopefully, this is it now. Hopefully everyone can see my slides now.
Rock and roll.
Good. So I'm just going to give you a quick update here today. I am trying to, just give a little bit of a background first about the vaccine development path, then continue on the development of COVID-19 vaccines, what kind of considerations we have to put there. Then I'll give a couple of highlights of the leading candidates, since we're hearing a lot of news. And then I'll even give you a very short update of what we're doing actually in my lab. So in terms of the vaccine development path, it's usually a relatively slow process. The conventional vaccines are based on inactivated or attenuated pathogens. So basically you first have to identify and isolate your pathogen. Okay, in this case, this was relatively quick. Then you have to grow it in culture. This was done quickly as well, since we do know that it is a coronavirus and how to grow those viruses. Now comes the complicated path with conventional vaccines. If you want to develop an attenuated pathogen, this can take years or decades. So this is something that led to vaccines such as the yellow fever vaccine that is still used. It is an old vaccine that was generated in the, I think, 1930s or 40s, and is still used pretty much the same way how it was generated back then. What is faster is a killed vaccine that you see here. That's your classic vaccine, like a flu vaccine or the inactivated polio, where basically the pathogen is killed by usually chemical treatment or radiation, and then you produce a vaccine. That's also relatively quick to make. The problem is that you have to grow large quantities of an infectious pathogen. And then, of course, we have all of the modern techniques, and that's what we'll be talking about mostly today. It is vaccine platforms that are actually based on molecular biology, where we first characterize what are the components of the vaccine what are the components of the pathogen that are required to generate immunity that will finally protect you. So as one example here, they're talking about here toxins. So toxin vaccines actually are something where recombinant technology has already made some progress. Let's say, for example, with anthrax vaccines, you can produce the protective antigen in a recombinant form and then immunize people with that without actually risking anyone getting sick. What people have done in the past with toxin vaccines was to generate toxoids where the toxin was detoxified with an antibody or with some other processes. We don't have to do that necessarily anymore now. Then, of course, you can clone those genes, you can purify, and these are now additional antigens that we will use to develop things like a protein subunit or DNA vaccine. So, just to summarize, conventional vaccines are inactivated or attenuated pathogens. Many classic vaccines are like that. Subunit vaccines, there is conventional ones where you just purify the subunit from the pathogen, or recombinant approaches. Recombinant approaches to make a subunit vaccine, and then other recombinant approaches that we'll talk about are DNA, RNA vaccines, or using viral vectors for antigen delivery. What's here, I mean, I'm just on this slide, I'm just giving a bunch of examples. So live attenuated, you have classic things like smallpox, polio, measles, mumps, rubella, and so on. Inactivated, purified, definitely influenza is a good candidate here, as is the inactivated polio. Live attenuated bacteria, wholesale bacteria. These are all conventional vaccines on the left side here, including the purified protein. This is now your subunit. That's the acellular pertussis vaccine that doesn't necessarily work that great, and that's why we have more modern approaches now. So next generation is purified virus-like particles that are used for hepatitis B vaccination or the papilloma virus vaccines. Purified polysaccharides that we can produce now also in culture in large quantity and purify highly, so pneumococcal vaccines, and then the more modern conjugates of polysaccharide vaccines. Again, this is, of course, just kind of to generalize here for the entire field of different vaccines. What we will be talking about today a lot will be plasmid DNA or RNA, as well as adenovirus DNA delivery, which is a recombinant viral vector that was actually originally developed for gene therapy. So just to give you an overview, different vaccine platforms have a different type of immunogenicity. And that is important depending on the pathogen that we try to protect against. So there's antibody-mediated responses. So killed virus vaccines are really good at that. So that's why the flu vaccine produces pretty nice titers that then we can read out with an assay. A very good vaccine is live attenuated. Yellow fever, again, as that ideal example. That does both antibodies and T-cell mediated immunity. And that's why you typically get lifelong immunity with a single shot. That's an ideal vaccine, but might take decades to produce. A conventional DNA vaccine is actually producing antigens in the muscle that is being injected with it. So you get pretty good T cell responses, but because those antigens are only produced in the cells and not necessarily properly presented, typically antibody responses are a problem. So you need to give very many shots, typically 3 or 4, to get a full course of immunity. Virus-like particles are basically looking like viruses, but they don't have the genome inside, so they cannot replicate. So that's actually very safe. They produce good antibodies, but it's kind of not ideal to generate T-cell immunity. And then pretty good are these single-round infectious particles. So this is now what we'll be talking about with the adenovirus vector platforms. These are viruses that look like a virus, that basically produce a high titer and have a incomplete genome inside that basically will allow the virus to make proteins as if it was a normal virus, but not replicate. Therefore, it infects one cell, but cannot escape. And then it looks to the body like that cell has experienced this viral infection. And that's what these modern vaccines are based on. And then this different type of immunity is now what we kind of have to look into. And then we have to look at the depressing generalized development path. So typically vaccine development takes 15 to 20 years, costs between $500 million and $1 billion. And takes, I mean, all of these different development steps. So what are we trying to do right now with the COVID vaccine? Basically, everybody is jumping in here and trying to do preclinical, so drug discovery part is kind of done because we know coronaviruses reasonably well. So now we're jumping into preclinical at the same time while we're trying to also do clinical trials. And this is what we're observing right now. The reality is that we cannot shortcut a lot of these steps, especially the clinical phases. And that's why, I mean, as you know, 2 phase 3 clinical trials started earlier this week, which is, of course, extremely fast, but this still will take many months to complete, and we don't know the outcome right now. That's just setting the stage. This is again here now the options that the FDA will have. So there's these red arrows here on the bottom. So for an emergency use, like for the COVID, there's an accelerated development review that will kind of go in parallel while the development is ongoing and the clinical trials are happening. And then actually already during the IND phase, it is possible that that an emergency use authorization can be given. That might be the most likely aspect of actually being able to start vaccinations sometime this year, if the leading candidates really deliver as promised. And that's, of course, mostly based on safety. So I'm going to just talk quickly right now about some development characteristics for COVID-19 vaccines. What's important here first is looking in the literature. And one of the things that has happened is that when people developed vaccines for SARS coronavirus or MERS coronavirus in the past, there was an unexpected and not so nice finding that basically there is this pulmonary immunopathology that could be found in mice or monkeys that were vaccinated and later on challenged. This was particularly with inactivated vaccines adjuvanted with alum, the standard adjuvant for vaccines. So this was then kind of termed antibody-dependent enhancement, even though it's not a classical antibody-dependent enhancement effect as you would see classically in dengue or so, other flaviviruses. And then basically this syndrome was even actually found in a re-challenge experiment. So what was seen that a monkey that had survived SARS challenge and was re-challenged actually potentially could develop an increased pathology. This just brings to the forefront that there is an effect that could be linked to a substandard quality of the immune response. Why do I say that? That's just basically setting the stage that there's a risk that we could develop a vaccine if we push that to the market very quickly, and the vaccine generates this this immunopathology subsequently when you get infected. Then this could be a real problem, because we already have issues with people that are against vaccination. And so this could turn into a real disaster. But now let's go at the work that we need to accomplish. The antigen that is of interest are typically membrane glycoproteins on the virus. Coronaviruses have 3 glycoproteins on the surface. Good that we know which one we need to look at. It's not the envelope protein, as it would be in flaviviruses. It is not the other membrane glycoprotein that's there, but it's the spike protein that makes these nice little kind of lollipop-like structures on the surface of the virus. This spike protein has 2 functions for the virus. One is receptor binding. So the virus needs that to be internalized into the cell. And then it also mediates membrane fusion. Those are 2 different steps. This is the structure of the protein, basically the amino acid, the structure of the different regions that you see here. And down on the bottom, you see the 3D structure that was determined with cryo-electron microscopy. The protein, spike protein, is a class 1 fusion protein, just like, for example, the HA protein for flu. Similar, the HIV GP120 or 140, as well as for filoviruses, the glycoprotein there. It exists as a trimeric protein, and it is metastable because it actually has to shift structurally to allow the receptor binding and then later on the membrane fusion. Has 2 different subunits, S1 and S2, and then they form this interwoven cap that you can see here. And actually there's a structural rearrangement so that cap can move up and down. And that movement actually is linked to the preparation for the membrane fusion, which is required later on, so that the viral RNA can enter the cells at the cytosol of the cell. So there's a pretty high homology that was found early on between SARS coronavirus and SARS coronavirus 2, which is the second one now, is the one causing COVID-19. The other one was the 2002-3 outbreak, mainly centered in Northern Asia and Canada. And there's a receptor binding domain on the S1 portion that actually binds to the cellular receptor, which in this case is human ACE2, which is a angiotensin converting enzyme that sits on most of the target cells. So one of the issues that happens is, this is here for the MERS coronavirus, the receptor binding and fusion is shown here. For MERS coronavirus, DPP4 is the receptor, but you could just envision that this is ACE2. So the protein will bind to it, kind of stretch out. And then actually prior to the fusion, a cleavage happens in the S2 portion. And that cleavage now allows this kind of spring-like structure to come up and grab now the second membrane. So, and this is really the the way how then this membrane fusion is driven by this fusion protein. And why am I showing you all of this? Well, we do need to stabilize the protein to not reach this post-fusion state. This is post-fusion. Post-fusion antibodies are the ones that are deemed to potentially generate this immunopathology later on. So in human convalescents, we actually see a greater reactivity to these post-fusion proteins, and they are not necessarily protective. And that's why we need a good vaccine, because natural infection may not protect an individual from reinfection with the COVID-19. Now we're just looking here at that receptor binding, and that is actually where what most vaccines are targeting. It is, as I said, the human ACE2 that is the cell surface receptor that the virus likes to bind to. And that receptor binding domain now interacts with that. And the only thing that is important on this slide really is that people have very early, this data is from, I think, March of this year, March or April, have very quickly been able to show that this receptor binding between this year's SARS coronavirus 2 and SARS coronavirus is pretty similar. However, it is, in this one study, it was shown a 10 to 20-fold difference. In this study down here, about a 4-fold difference. So in general, there's a higher affinity of the SARS coronavirus 2 to the ACE2 receptor. And this may actually be the cause for this much greater spread of the infection. It was easier to control SARS coronavirus back when, because number one, there was more pathology in the patients seen. And then the second aspect was that the virus was not as highly infectious. So this virus has an evolutionary benefit over the original SARS virus. So now just quickly summarizing what the different approaches are that are being used to develop COVID vaccines right now. We start with life attenuated. I already explained, you would put that in a target cell. It replicates, will release. You have now your antigen here will go to antigen presenting cells and generate your CD8, CD4 immunity and B cells, and then hopefully generate some memory cells so that when we experience another infection that we are protected. So there's, of course, inactivated antigens that could be used here. Then the more modern approaches, the recombinant approaches, are either based on DNA or RNA. So with the RNA, of course, we put in the message that the cell is used to making proteins. When we use DNA, we actually have to drive it with a promoter. There's more risks with that, and we need to have a special delivery technique. Right now, electroporation is the one that is mostly used to deliver these kind of vaccines. For RNA, people have developed these lipid nanoparticles that seem to help to very quickly and efficiently shuttle that into cells. So all you need to do is just inject. Now, of course, there's these viral vectors that I spoke about. There's active replication, or you could also inactivate your viral vector. And then basically you can deliver a vaccine that is chimeric and will carry the spike protein for the coronavirus while also still kind of having the other host viral proteins there. VLPs are being used, recombinant subunits. We've talked about that. And this is basically now kind of summarizing all of the different weapons that we have. And I'll give you now kind of an overview of what the candidates are. There's close to 200 that have been known as of yesterday on this website. You can always check where vaccines and therapeutics are in terms of development. There's currently 13 inactivated, 4 live attenuated candidates in development, 23 non-replicating virally vectored, 18 replicating, 16 DNA vaccines, 27 RNA vaccines, 63 based on recombinant proteins, and 15 VLPs. Actually, I didn't update the number here for passive immunization, but there's also, as you know, many efforts ongoing for convalescent serum and antibody therapy, mainly based on monoclonal antibodies raised, as well as cocktails of those, and then convalescent serum treatment. In terms of the clinical status, this is where we are. So there's currently 4 vaccines in phase 3 clinical trials. 1 inactivated that is being developed in China, then 2 RNA vaccines that are both, I think, in phase 3 clinical trials in the US now, and then a non-replicating viral vector. This is the Oxford vaccine that is mostly tested in the UK right now, but I think also getting into the US. You see some vaccines in phase 2, a few also in phase 1, and then a lot of them just in preclinical stage. But it is amazing that there are actually so many in the clinic already. So I'm just going to give you very quick highlights here of where the leading candidates are. Of course, everybody has heard of this company called Moderna that is developing an RNA vaccine candidate together with the Vaccine Research Center. This is a timeline that they have in one of their preprint papers, where they're basically showing that, you know, just like 2 weeks after the first notice of the virus popping up in China, the sequences were published. And then just a few days after that, Vaccine Research Center and Moderna already started to develop prototype RNA vaccine candidates. And then they started a clinical trial just 2 months after that. Why is that they can be so quick? Well, manufacturing RNA is a chemical process. Therefore, as soon as you know the sequence, you can make it. And you can also have pre-approved FDA protocols in place that will allow you to put your RNA vaccine into humans, because you're always delivering the same material. You have a certain safety file already. So that is one of those benefits. So this is some of the data that they have, interestingly, still only in a pre-publication. This is mouse data. Typically, mouse data is the first one that's getting published, but this is not out in a peer-reviewed publication yet. But what you can see is that they did a dose range in BALB/C mice, C57, and then those B6, C3. So they showed that in various immune environments, you see a dose response. And most importantly, you can raise neutralizing antibody responses as well. Moderna has also, as I showed you, gone into a phase 1 human clinical trial. Of course, one of the most important things here is safety. So they tested 3 different doses, 25 micrograms, 100 microgram, and 250 microgram. If you remember, the highest dose in the mouse was 1 microgram. So that's really, it's quite a difference in the range. But you'd need a little more in humans. I would say that we have to, still be very cautious, because when you see after the first vaccination, there's already some symptoms, especially, of course, some injection site pain, but particularly after the second dose, which is expected because now the body will react to it, particularly at the highest dose level, the 250 microgram, there have been quite a number of severe reactions all throughout the different conditions. That is not necessarily a problem, but it may limit the population that can be vaccinated with this. Based on this data, they said to go forward with the 100 microgram dose level. They actually, I think in phase 2, were testing 50 and 100 microgram dose level, might even be in phase 3. I'm not 100% sure right now what doses they're using in there. of course, what's now important is the immunogenicity. So immunogenicity was pretty good. After a single dose, as you can see, after 15 days, there's seroconversion in pretty much all the individuals. Probably the background is not too high, but then they gave a second dose, which kind of solidified the response. This is the response to the whole, this is the pre-fusion stabilized spike protein. As I told you, this is what we want to get a response against. And this is now the response against the receptor binding domain. If we inhibit receptor binding, the virus cannot get into the cells. So this is the most important functional response. And after one dose, I would not necessarily say that this is very strong titers, but after 2 doses, it is very good. And 100 microgram and 250 microgram dose levels were pretty good. What they're showing here is convalescent serum on the side. So these are the responses that a natural infection will cause. And you see it is lower here for the receptor binding domain than it is on the left for the spike protein, which has to do with some of those effects that I mentioned earlier. And then down here are neutralizing responses. Neutralization was not very good after the first dose, but it was very potent after second dose, again, but especially with the 100 microgram dose. And you can see this against wild type virus here on the side. This is the antibody kinetics now in Rhesus macaques. The Rhesus paper was just released this Tuesday on the 28th. And what you see in here is they were doing 2 different dose levels, 10 and 100 microgram doses in Rhesus macaques. And you see also very good immune responses binding IgG to spike protein, and then virus neutralization. There's a dose response, blue being the low dose, red being the high dose. So definitely the high dose is better. The 10 microgram does not seem to be that wonderful. And then down here are the titers for individual animals. You see that it is relatively consistent. This was 8 animals per condition. So it's a relatively large N for non-human primate studies run so quickly. And just for comparison, again, you see convalescent serum titers here from humans. So they're much lower. What they also showed is that there's a good activation of Th1 type cell-mediated immunity here in reactivation of memory CD4 cells and not so much Th2. Again, this indicates that it would not kind of drift in that direction of immunopathology. Same thing here for T follicular helper cells, but I don't want to bore you too much with those research details. What is most important here is that these monkeys were challenged. They were challenged by intranasal and intratracheal instillation with virus. And what you see here is in your controls, there's good viral replication. This is in bowel fluid, so this is lower respiratory tract. You see infection in most animals, and then, you know, going down after 4 and 7 days because the virus does not make most of these animals sick. What is interesting is that at the low and the high dose level, protection in the lower respiratory tract was almost complete. However, when we look in the nasal tract, now there's a differentiation. So you see that the 10 microgram dose did not protect about half of the animals, but at the 100 microgram dose level, there was a pretty good level of protection. What does that mean? As Dr. Brostom said earlier, there's 2 different qualities of a vaccine. One is to protect the individual, and one also to protect the population. So a vaccine that will protect us from replication in the nasal mucus will definitely have a bigger impact on the further spread of SARS coronavirus 2. So basically, they've shown good immunogenicity with 2 doses, seems a very good balanced response, potent virus neutralization, and this reduction of viral loads in the Rhesus model, both in upper and lower respiratory tract. questions that still remain are durability of immunity, adverse reactions, and the exclusion criteria, so who all can actually receive this vaccine. And then also the question, what would be efficacy after only one dose? And what are the kinetics of the immune response? Because you saw that the 2 doses are pretty well needed. Next up is the...
Dr. Axel, I'm going to interrupt you just really quickly. Do you know if, and I apologize, do you know if that test about the length of immunogenicity, which goes beyond just measuring IgG and IgM, because it may require T cells. I'm just wondering if you know that they're going to test this with all the pressure to go so fast. Are we going to have a cadre of monkeys that are immunized that are going to be exposed 2 months down the road rather than in sort of a compressed time frame? Over.
Yeah, I think that their paper that came out this week was somewhat alluding to that they are, or that they have already started a durability experiment. I don't know for how long. I think there was a talk about a year, which would be very appropriate. I think that would be a good durability. So they are looking at that. And I will say that, I mean, that is really one of the things where I trust the Vaccine Research Center a lot. It is great that they are involved in that it is not just a company pushing this. Vaccine Research Center really carefully looks into that, of course, because they have a long history in especially HIV vaccines, and we all know what the issues there are. So I believe that this is being looked at, but however, with all of the pressure that we're undergoing, I don't see that these studies will be completed before products will be rolled out. So that will be that caveat that I will have to put there.
Yeah, and in addition, you did mention that the Th2 was not so activated, and then you alluded that sometimes indicates that this this immune response to being vaccinated, particularly for those who might have been who've had SARS-CoV or COVID-19 already, that this looks like it might be okay, and that may not be such a bad side effect. Over.
Yeah, so basically the biggest problem that, I mean, so this is now, again, just based on the literature generated with SARS and MERS. So the indication there was that incomplete protection or immunopathology may be linked to a higher upregulation of Th2 immunity, where you produce antibodies that are not necessarily protective. You're basically deflecting the immune response. This, I always, compare that to, it's basically like, somebody, where you kind of hear a melody and you think, oh, I got it now. And then, but, because you have a terrible memory, maybe and you can't replicate that melody now, that song kind of is not quite the right one. And that's what our immune system will do. This is how we get allergies and things like that. So we basically activate the wrong component. And that's what I think this indication here is that the immune response is triggered in the right direction. However, we're looking here at memory cells, but this is only 4 weeks after the second dose. So we don't have any idea right now how that looks 3 months down the line, 6 months down the line. And those are those bigger questions, I think, that you're absolutely right to ask that. So it's real important.
Thank you.
Okay, here we have now, this is the Ad5. So adenovirus 5 is a common cold virus, very benign in a normal infection. Let's just get right to the problem. Ad5 circulates pretty widely, especially in Western countries. So there's a high level of seroprevalence, and if you have good neutralizing titers against Ad5, this vaccine will probably not work. So the platform, however, is very attractive because it is a very potent immunogen. It is replication incompetent. So this is now a viral vector that will express the spike protein, and it will just infect that one cell. As you can see, very high amounts of these viral particles, 1 times 10 to the 11 or 5 times 10 to the 10 have to be used. The main reason is because you only have that one chance, because now you are going to generate vector immunity in the person, and you will not be able to revaccinate with this for at least 6 months or maybe a year. So you have to have protection after a single shot. That's why they use these very high titers. It's promising. They show pretty good here ELISA binding, ELISA antibodies binding to the receptor binding domain, of the spike protein. And then down here, virus neutralization. This is also a phase 1 human clinical trial. Safety was pretty reasonable here. And then, so this is CanSinobiologic that develops this. So there's that problem of the pre-existing immunity to the vector. And now we come to the Oxford vaccine that everybody has heard about. So that one also is in human clinical testing, commercially being developed by AstraZeneca. And of course, they have gotten quite a bit of money to develop that already. Here are some, safety results. So this is a vaccine based on a chimpanzee adenovirus that the human population is not pre-exposed to, and you don't get so high responses against the vector. So you can actually give 2 doses. More than 2 doses within a short period does not work, but typically a 0-4 week regimen is possible here. As you can see, typical side reactions, it is a viral infection that happens, again, just a single cycle. Safety should be relatively good. I mean, there's a similar vaccine that has been developed against Ebola that is kind of in very advanced stages of development as well. And it has been used also in immunocompromised people, but there will be exclusion criteria here for sure. What is important is that when you look at the prime and boost response, this is a single dose here. You see you get good titers, but it isn't quite stable and it's a quite wide range. So that's why they did the prime boost here in the second group. And there you see that it stabilizes in a very similar range as the convalescent serum samples. As you may remember, the Moderna results were quite a bit above. Of course, these are different assays, different companies, but it's just kind of a point of reference when we're trying to understand how far developed certain candidates are. These are virus neutralizing titers. So as you can see, after the first dose, virus neutralization is not that great. So you do need a booster vaccination to really be protected. And then the dose range is, again, pretty similar to convalescent humans. This vaccine has also been tested in Rhesus macaques, and they showed that there was no reduction of the viral loads in the upper and I'm not entirely sure, maybe not even in the lower respiratory tract. However, there was no extended amount of viremia, and it was deemed that potentially protective. This is the cell-mediated immunity, since we were talking about that earlier on the Moderna vaccine. So what you see here is after a first dose, actually pretty good cell-mediated immunity is up already. And this is one of the things where these virally vectored platforms will probably excel better than other platforms, because the recombinant virus infects the cell, and then as it is associated with the cell, that is what raises potent cell-mediated immunity. And so that's one of the highlights for this. And then I just wanted to give you a few updates of what we do in my lab. This is just from a paper that we pre-published last week. I don't do that typically, but right now this is en vogue. So we're trying to follow the game. And this was based on a platform that we have developed for filovirus vaccines. And so the most important point in this experiment was only to select what adjuvant to use. So far in the other platforms that I've shown you, adjuvants did not come into play. The adjuvant and the production of antigen are things that do take a little bit longer for recombinant subunit vaccines. However, we have so many of these vaccines on the market that we understand immune responses better, we understand manufacturing better, and ultimately, when we want to roll it out to billions of people, it may be more rational because we have just this body of evidence. So what I'm showing here is just, this is after 2 doses in mice. You can see that we have a potent immunogenicity with all 3 vaccines. Most importantly, when we use our lead adjuvant, this is the one that we use in our Ebola virus vaccine. We have actually very high titers already after a single dose. But this recombinant subunit vaccine. The other 2 catch up to that. One of them is adjuvanted with alum, which of course is used in many vaccines that are in human use. However, the Covaxin is a clinical candidate, so it has been in human clinical trials, so we believe that this could be advanced very quickly. What's very important as well is that this vaccine with our Covaxin adjuvant raises potent, also cross-neutralizing, cross-reactive titers with SARS coronavirus. So it may actually be a group-specific one. So if there's an antigenic drift over the years, you know, it may have a little more broad specificity. But then most importantly, the Covaxin adjuvanted formulation after 2 doses gave us very potent virus neutralization titers. And that is really key. And then we were talking about T-cell immunity before. So we can see actually these are spot forming units in an ELISA, in an ELISpot format. And so these are also cells that are secreting interferon gamma. So they're Th1 type responses. We see the same thing as Moderna, where there's no activation really of a Th2 type cell-mediated immunity. So we have potent humoral responses with good virus specificity with our lead adjuvant, high virus neutralizing antibody titers, and then potent induction of a Th1 type immunity. Where we are going here in the future will be next testing a thermal stabilization method. This is something that I'm sure everyone out there on smaller islands than ours here will be very interested in, because we're trying to have a thermal stable vaccine that will be lyophilized in a vial that you just need to reconstitute with water for injection, and then you're good to go. We need to, of course, determine optimal antigen adjuvant dose and then a fully scalable production method, which is something that we do in collaboration with Hawaii Biotech, also here in Honolulu. We then will finalize a thermal stabilization method so that we can actually fill those vials, define correlates of protection that will be using mouse and non-human primate work, as well as manufactured and clinical grade material. Hopefully, we will find funding that we can actually advance this product into human clinical trials very rapidly. With that, I would just like to acknowledge my team. I have a very hardworking team, despite all the COVID shutdown and whatever. We've been working really hard over the last several months, and my collaborators at Hawaii Biotech, BioQual, and Sologenics have also helped us, as well as MAP, with providing some antibodies. With that,
thank you, Dr. Lair. That was remarkable. I mean, I think for a few minutes there with the immunology slides, it was close to, you know, I had a flashback to 1988 in North Carolina, sitting in my medical school lecture But then you brought us home and listed those vaccine candidates that are in phase 2, phase 3 trials. It's pretty clear we're going to see one of them. Something's going to have to get selected. At least I would hope that we're going to continue to have a successful launch of a vaccine. And as you mentioned, I think that the thermal stabilization will be a huge issue for us. I remember being in Sierra Leone when the vaccine was first being tested, and the temperature requirements were so extreme for this experimental vaccine that it was almost impossible to use where we really needed it the most. So hopefully this will have some thermal stabilization at the time it's released so that we can use it quickly for the region. Are there other questions for Dr. Lay? We have about 5 minutes for questions, and then we need to start in right away with our lab.
So let me just add you one thing here, and that's just because you were talking about thermal stability. So what we've shown with our Ebola and other filovirus candidates is stability up to 12 weeks at 40 degrees Celsius, but no decline in immunogenicity.
That's plus 12, not minus 12.
It's 40 degrees and then 12 weeks storage. So
12 weeks to 40 degrees. That's great. That's doable.
That's African ambient temperature. So
yeah, we can do that. That's amazing.
Or South Pacific.
Yes. Other questions?
This is Shai.
Yes, please go ahead.
Just 3 simple questions from my group here. There are so many of us, but this is Dr. Skilling. I'm sitting next to the monitor and the microphone. So first one is from Dr. Bass, our OBGYN. She would like to know if the vaccine can be used in pregnant women.
That will depend on the candidate. I would say the virally vectored vaccines, not. The mRNA may be. Our vaccine, the recombinant subunit, most likely can be used. We've been doing experiments in pregnant mice. So we have a bit of experience already in studying that in pregnancy.
Thank you so much. And then I just wanted to thank you for thinking about us with the thermostable vaccines, because you know we would need that. And my question is mostly for the community educators. You're talking about one vaccine might be out before the year of observation in non-human is completed. You know how messages go out on Facebook, the community will read it, and they're gonna ask us, why vaccinate us now when you know you haven't gotten the full year of observation, and then we're afraid to take that vaccine. What's your advice for us to, when we talk to the public, the community? Thank you. Out.
Yeah, I think the most important thing will be really, I mean, we need to monitor. This is something so rapidly developing, you know, ask me again in 3 months or so, you know, but right now I would say that we have to educate the community that vaccination will be the only way that we will get out of this current situation. Because as you have seen, the even natural infection may not protect you. Whereas we do know that a good vaccine will protect. However, I will always also say that you need to be absolutely sure that the safety profiles at the least are met. And then, you know, the durability studies, hopefully they will at least show an interim, like a 3-month time point or something that, you know, will then build that confidence.
Great, thanks, Dr. Lay. We have a question from Chuk team. Please go ahead.
Greetings. It's not a question, actually making a comment on this. I heard there are only 3 vaccines on the line, has been in phase 3, but today's news is that one has been limitedly approved for use, that is CanSinobiologic in China. And in that vaccine war, actually, how soon we can get one vaccine for commercial use? What is the timeline on that in terms of its immunogenicity, thermal stabilization, particularly cost and availability. What is your focus? What is your view on that? How soon we can get a commercial vaccine for usage? Over to you.
Well, I think I was hopefully showing you a little bit about the technology because I had the CanSinoBio on here as an example. This is this Ad5 based one where, you know, the concern is that people that have previously experienced adenovirus 5 may not respond to the vaccine. So that's one of the issues. Thermostabilization is possible for adenoviruses. So technically that could be done, but I don't think anyone has done this yet for this vaccine. So if you need thermal stability, this will take some time. But then ultimately it will depend on CanSinoBio, how their marketing plan, you know, works. I would be very cautious. Adenovirally vectored vaccines, you know, are not necessarily, I mean, they're very experimental. There's none that is approved for clinical use. There's a partial approval, I think, by the EMA now for a adenoviral Ebola virus vaccine. But, you know, I would, you know, cautiously still, you know, watch the data, because one of the worst things that could happen is to go too quickly and then have to pull back and say, well, sorry, now that vaccine doesn't work so well. Then we have the problem with the anti-vax. I mean, that's now where the Facebook and whatever messages will go flying.
And I suppose it's possible that vaccines would be available for islands to purchase on their own from, you know, whichever one you choose, and could be your favorite vaccine from your favorite country, who knows. But my guess is that this would probably come from WHO, from a larger committee, or from CDC, who supplies you with your VFC program at present for all the US, northern US Pacific Islands. So it may not be an option on the open market for any of the islands. I just don't know how this is going to be marketed going forward. Any comment on that, Dr. Lair?
I fully agree with you. I think there will be guidance coming from WHO, CDC, and one of the things, of course, that we have to keep in mind is that politics also plays a role. And like you mentioned, there's a possibility that China may choose to, you know, market to certain aspects of the Pacific region, as they are trying to gain influence. And I mean, there's nothing wrong per se in this. I would just say what is important is that we need to look at the data. We need to have solid data, because you don't want to go out and later on have to explain that, well, we rolled out a vaccine maybe a little bit too early.
Okay, thank you. And I really love that question from Coach Ryan and Secretary Skeeling about preparing our community, trying to do battle with the misinformation that flies around so quickly across every community in the world. So it's always hard to battle misinformation for a vaccine. For this one, it might be you know, doubly difficult as we move to try to protect our populations. We'll just have to see. Dr. Lair, that is fantastic. Thank you very much for each moment of that. Again, it's recorded, and other folks will, if you know folks who weren't able to log in from the Pacific communities, this will be available on the PIHOA website. I apologize. Let's move on to the next talk. And prior to doing that, I want to challenge you guys. Please either leave your email and your name in the chat box so we can count you, or if possible, you can send me an email later, and so that we'll know who actually is logged on. CDC is asking us to try to keep track of the great audience that we have for these talks. All right, so let's move on. I'm a little nervous here, Dr. Chan, but let's give it the old try, okay? I'm going to stop share, and please feel free to get us started with the lab section. Unfortunately, we still don't have any sound. Dr. Chan, we're unable to hear you. Let me see if I can check and see if you're automatically, no, you're not muted from my side. Sometimes, Dr. Chan, you'll have to go over to the microphone icon in the lower left, and you will want to click on that little caret, and that will give you several options for your microphone. And sometimes that makes a big difference for us. I think he's going to call in again. Please be patient. This is where it'd be helpful if I knew any good jokes. Or
Hello?
could sing. Yes, we can hear you.
Okay, I apologize for that.
No problem. Very happy to hear you.
Gosh, okay, let me try again. Is it, can you see me now?
We can see your slides. Yep, it's better if you click on the presentation view.
Yes.
And now go over to display. Now go up to display settings. There you go. Perfect. You're up.
All right.
Great. We can hear you fine and see your slides. Thank you.
Great. I sincerely apologize, everybody. As you can see, I am not an IT expert. So thank you for your invitation, and again, I apologize. Now I think we're okay now. Okay, so I have a short talk, and it's about laboratory testing and for COVID-19. And it's going to be relatively short. Before I start, I always do the disclosure. I'm a pathologist. I'm affiliated with a group called Pacific Pathologists and Clinical Labs of Hawaii. So I'm part of a laboratory company. I'm going to talk about tests, but these tests, we run one of the tests, but I don't have any personal financial stake in them. Okay. So the purpose.
Dr. Chan, we can hear you, but your sound is coming in and out a little bit. If you could get a little closer to the mic, it might be better. Thank you.
Okay, how's that? Okay, so the goals of this talk is that I will talk about the different types of tests for SARS-CoV-2, and basically we'll talk about test principles, basically how do these tests work, and test characteristics.
Sorry, Dr. Chan, it was actually better before with the prior method.
How about now? Oh,
it's perfect.
Okay. So I'll talk about how these tests work and how good these tests are. Okay. But before we do that, we should do a timeline and look at it, and it'll give it an understanding of how things have transpired. So back in late December, if you all remember, back in 2019, an outbreak happened in China, and it was a pneumonia of unknown cause occurred in Wuhan, Hubei province in China. And then the WHO reported that Chinese authorities determined that the outbreak was caused by a novel coronavirus. And the CDC on January 21st confirmed that it was confirmed the first evidence of U.S. Infection in Washington State. So, and subsequently, February 4th, the U.S. HHS determined that this was a public health issue, and the CDC developed a diagnostic test and was granted an EUA, or emergency use authorization. And so, as you all know, normally tests take several years to be FDA approved. So this is not your typical approval. This is an emergency use authorization. So the review that these tests undergo are expedited, and it's not your typical approval. So just understand that going forward with all these tests that we have now. So back in February 11th, the virus was named SARS-CoV-2, and the disease was named as COVID-19. And then this date on February 29th is important because this is when manufacturers, so non-governmental agencies, could develop tests for SARS-CoV-2. And then at this point, then all these manufacturers could produce tests that then that we have today. And just as information, on March 10th, we started testing for SARS-CoV-2. This is offered just a couple of weeks, and this is unprecedented because usually when you bring a test on, it takes months. So we were able to bring it out in weeks. And then so bringing it on to our present day, so today and tomorrow, there are currently more than 150 manufactured tests for SARS-CoV-2 that have this FDA emergency use authorization. So they're all different. And then looking at this slide, it's good that Dr. Lara went first because he went through all of this. But this is, as you know, this is a picture of the virus, SARS-CoV-2. And illustrated here are the proteins on the surface. And so the right side is a the virus, and you see the proteins on the outside and the internal RNA that comprises this. So the point of this slide is just to show you that the virus is made up of different proteins and different nucleic acid, and that's important because this is what the laboratory tests are based on. So this slide shows tests grouped in 3 different categories. And this is kind of the way that you should envision what the tests are. So I'll go over them in detail. The first on the top left is nucleic acid amplification testing, so NAT. And when you hear molecular, this is what they mean. And this is an umbrella term. And this kind of testing detects the viral nucleic acid, so the stuff in the middle here in this virus. And the specimen types you would use are nasopharyngeal, nasal, oropharynx, because that's where all the viruses are. And that's where you collect them. Moving down to the bottom left, we have immunoassays, and these assays test for the protein of the virus. So if you imagine in this picture, it's the yellow or the light blue. And again, this test detects actual virus, but instead of nucleic acid, this is protein. And it's the same specimen types as the NAT testing, because it's essentially you're looking for the actual viruses. Then, okay, when we move on to our third category on the bottom right, serology. And this this type of testing detects antibodies. So it's actually not the virus itself. And it's detecting how your immune system reacts to the virus. And this kind of testing is not used to make diagnoses of active infection. Rather, it's used to determine if you've been exposed or had prior exposure to SARS-CoV-2. And so therefore, the specimen type will be blood, because that's where the antibodies are primarily in. And so it's would not be nasopharyngeal, nasopharynx, etc. So this is kind of the three categories that testing makes up. And then so we'll go into each of these a little bit more detail. So nucleic acid amplification testing is, again, a broad umbrella term, and it's also known as molecular. So you hear that term. And from a very basic method, the way you do it is you extract the nucleic acid from the virus, you amplify it, and you detect it. And so those are the three basic steps of doing NATs. So more specifically,
Sorry, Dr. Chan. Again, we're having a little bit of problem with the audio. Sometimes it's just amazing. It might be that you're coming closer and farther away from the screen. Please continue to try. Thank you.
Sure. Okay. Is this good?
Yes, really good.
Okay. So NATs have different types of technology, or it could be subdivided. And the more commonly known types of NATs are PCRs, or polymerase chain reactions. And of the polymerase chain reactions, PCRs, there are different types of there are different flavors of them. And then this particular testing, it uses reverse transcriptase PCR, and it's real time. So those are just qualifiers that pertain to PCRs that use viral RNA viruses, RNA from PCR for viruses. So this technique uses reverse transcriptase, converts RNA to DNA, and then amplifies several nucleic acid regions of the virus, and that's shown by this diagram on the right side. If these polymers are specific for certain regions within the virus, and it has these fluorochromes on them that give off a light when you shine a laser on it. And this PCR is based on multiple heating and cooling cycles. So you have to do many of them, and that's why it's called a polymerase chain reaction. It's several cycles of this, and as viral regions are amplified, fluorochromes are liberated, and they have the ability to give off light when you shine on them. So that's what I'm showing here on the left, with uses fluorescent probes to detect reaction in real time. So this is happening in real time as the cycles are being heated and cooled. What I'm showing here is a graph of what the signal looks like in real time. So the number of cycles are shown on the x-axis. As you go in time, fluorescence goes up if there's virus that's amplified, and it crosses a certain threshold. This is shown by this dotted line here, and that's called the CT, or cycle threshold. And you'll hear those terms when you talk about this kind of testing, like what's the CT? So that's the lower number CT you have, it indicates you have a higher amount of virus, because you can imagine if you have an original high number of virus to begin with, as you start amplifying, it'll amplify faster, and then it'll cross this threshold. Whereas if you had a lower amount, it'll cross later. So a high CT value indicates a high viral burden. A low CT value will indicate a low viral burden. All right, so that's RT-PCR. So there are other types of NATs, and in this case, there's isothermal nucleic acid amplification, and further subdividing that, you can have a type of isothermal nucleic acid amplification called transcription-mediated amplification. The terms here are not that important, just rather know that these are different types of molecular tests under this NAT category. So the difference with isothermal nucleic acid amplification is that it's held at a constant temperature. There's no cycling, there's no heating, cooling. And essentially it's the same principles, because you use fluorescence as well. After a certain point, you'll have enough fluorescence if the virus is present, and you read it out. So there are some positives to this. It requires less energy and potentially can be faster than PCR. So this is just kind of terms and technology that people who are in the know would be interested in, and this is just for your understanding how the gnats are subdivided. This graph here gives you a picture of what the virus, if you were to lay it out in a single line, and you've seen this in Dr. Lara's presentation. This is the virus with their different coding regions. And what I'm showing you here on the bottom are the different assays, and this is not a comprehensive list. This is just all the different primer pairs that amplify certain regions. So in this case, this assay would amplify the ORF1b region, whereas this assay would amplify the N region. And the point here is that the molecular tests are not all the same. They use the same principle, but they're not all the same. And because they target different parts of the viral genome. So this may give one test an advantage or another, or disadvantage. All right, so this photo just kind of give you an idea what the instruments look like. So it's not as abstract. The testing that's done by the CDC developed test is run on instrumentation that looks like this. This is the thermocycler here. This photo in the bottom middle is the Cepheid line of products. I'm not sure if you probably have seen these. They're probably in your laboratories, and I'm going to assume it's the bigger product here. Bottom right here is the Roche instrument, and the top right is the Panther from ChAdox, and then the top here in the middle is the Abbott ID Now. So it's a small toaster-sized instrument. So versus the Roche, which is a big, probably bigger than a refrigerator. So it's just they're big and small. And the Abbott ID Now uses isothermal nucleic acid amplification. The CDC assay is RT-PCR. So they're all different types of gnats. Okay, I'll move on to the next category, antigen. So again, this is where the testing detects viral protein and not nucleic acid. And at least right now, we have two FDA EUA platforms, one from ChAdox and one from BD, and this is what they look like. You can see it's kind of a small, almost like a pregnancy test cartridge. You put in the sample and you just put it into this instrument, and in a couple of minutes you get a result. And the way this works is through this cartoon. So we have here a test strip that have antibodies to SARS-CoV-2 virus. And so here's the virus shown here on the left. You mix it with other antibodies that are hooked up to fluorochrome, and then you run all of this onto that cartridge. And then so you have this complex here. And then in the instrument, light is shined, and then fluorescence is given off, and then this is measured by the detector. So if no virus, you should not have any fluorescence. If you have a virus, you should have fluorescence, and then the detection. Okay, moving on to our last category, antibody. So again, this is a test that looks at your immune response to the virus and not the actual virus itself. And the way this works is that it also is kind of like it is an immunoassay, and it captures the antibodies you make to viral proteins, and they come in different shapes and sizes. So essentially, you have a phlebotomy type where you draw blood and you put them on these big instruments that look like these on the left here, or you have a finger stick. This point of care type platform here, and it's shown on the top right side, it's essentially like a pregnancy test. You see one band or two bands to see if you're positive. The phlebotomy type have better sensitivity and specificity than the finger stick types in general. All right, and the way these work, I'll talk about one particular assay. So this is the Abbott Architect assay. They have a particle. So this is not a virus. This is a paramagnetic particle, basically made of iron and their proprietary materials. They hook on the SARS-CoV-2 protein, and in this case, they use the nucleocapsid protein. So then if you apply blood from a patient, if that patient had a prior infection, he or she would have had antibodies. And so these antibodies would then bind to the nucleocapsid protein. Then you combat this complex with an anti-human IgG that's hooked up to a fluorochrome. You shine a light, gives off fluorescence, and it's detected in a detector and measured. And so if you have antibodies, you should have this complex to generate a signal. If you don't have antibodies, you shouldn't make this complex, and you should not make a fluorescent signal. So that's how the three different categories work. And so how good are they? And so this busy slide kind of breaks that down. So what I have on the top here are the molecular tests, and the bottom is the immunoassay or antigen type tests. And so what I have here on the left is basically the different metrics. You know, how many, let's say if you had 24 hours and you just ran everything nonstop, what's your theoretical throughput? And then what's your time to result? And then what's your positive % agreement, which is essentially like a sensitivity measure. And then negative % agreement, which is like your specificity measure. And then limit of detection, how many viral copies per ml can you detect? So the lower the LOD, the better your test is. And so what I have here are the different, a selection of some tests, not all by any means, and what I could find in their product inserts or the current literature. And so using the CDC test as kind of like a benchmark, if you ran that test nonstop 24 hours a day, you probably could get 300 patients through. And they reported a 100% sensitivity and 100% specificity. Their limit of detection was 85, and some other sources say it went up to 550. So whatever I found in the literature and their product insert, I just put it there in a range. So that's the CDC assay. Hologic, Aptima, higher throughput. That's the test that we use in one of our major labs here in Hawaii. Faster, a little bit faster throughput. Here, really good sensitivity, 94 to 100, and good specificity. The LOD is 500 to 5500. And so we just move on. So I see that several of your labs have the Cepheid, and again, in a theoretical world, you probably could do up to 400. It depends on the type of platform you have. And it's a fast test, 45 minutes, pretty good sensitivity and pretty good specificity with a good low limit of detection. The Abbott ID now has a lower throughput. It's a one-time, one specimen per roughly 4 to 20 minutes. Depends on how long you keep it in, because it's probably it'll pop up in 4 minutes. But if it's negative, you have to let it run all the way through. Different groups have reported varying sensitivity. So I just basically put this down for your perusal. And it was claimed to be a sensitivity of 73 or 74 to up to 100. So it's a bigger range. And the limits of detection are shown here. So the summary for the NATs is there's a large variety of how fast these tests are run, and you can see a range in the sensitivities and specificities.
Dr. Chan, just
so then comparing,
things.
Yes.
One is that in terms of the Cepheid expert, most of the jurisdictions have 4 channels. They may have 2 separate 4 channels, so their throughput is more like 100 possibly a little higher, but which is still a nice throughput. Very challenging considering that everyone has to be swabbed. The Abbott ID Now, I just wanted to also comment on that. The low end was the 73%, that or 74, which was reported out of New York City among a special group of folks, and resulted in a change also in the technique now, so that viral transport medium isn't used and people are swabbed and then directly detected, which means you can't do you can't do pooled specimens, but it probably has a better and slightly more closer to Cepheid when it's used without VTM. We're finding in the region that it seems to be identifying quite well. In fact, there's been a couple of folks that were positive on the Abbott that were negative on the Cepheid, but our experience in general is that both are working quite well and are comparing to the RT-PCRs that we have, which are ABI 7500s in general. And is that, when I see the CDC there, do you know what instrument that is?
It's ABI 7500.
Yeah, so that would be more comparable to that, which is still the gold standard. There actually are a couple of others that have been donated from South Korea or Taiwan in the region that are about to be used or are already being used. And they've also, in general, given quite reliable results. Over, back to you.
That's a fair assessment, right? So I use this positive percent agreement based on what I could find, but you're correct. They did modify their protocol, which hopefully improve their sensitivity. Thank you. That's a very fair statement. Okay, and then just using that same analysis, looking at the antigen testing, the two different assays that exist here on the bottom. Throughput is not as high, but it's fast. It's very 15 to 20 minutes. The sensitivity is not as high, and I'll show you. Initially, Quidel reported a 80% sensitivity, and then they recently released a bulletin saying that they redid their testing, their study, and then they said it now is 96.7%. This needs to be confirmed by the community researchers that are non-Quidel testing. So just keep that in mind. In general, these immunoassays tend to have a lower sensitivity, and 80% seems about right. So I'm optimistic that if it works, great, but it needs to be confirmed. Again, the value of this testing for the antigen is that it's fast and it's easy to do. So I've shown a couple pros and cons here, and it's just, it could be placed in a variety of settings.
Dr. Chen, one other question about the antigen testing, if I may, and that is, if we know that the molecular testing suffers from sometimes an overdiagnosis of people post-disease who are still having viral particles, non-infectious positive cases that are confusing lots of jurisdictions, but everyone's kind of figuring it out and going forward. Do you know, does the antigen test suffer from the same problem where there's viral particles that would trigger a positive result in someone who's actually well past their infectious period and asymptomatic? Over.
Right, that's a good question, and the answer is I don't know, but I would imagine less so. So as Dr. Brushram is mentioning, what the NAT test are nucleic acid, and so let's say if you have the infection and then get over it, you still may have nucleic acids still around, but these are non-viable viruses. But the NATs detect the viruses whether they are viable or not, whereas the immunoassay is looking for proteins. And I imagine the proteins don't stick around as long as a nucleic acid does. So I would assume that you wouldn't have that problem like the gnats. So the gnats are really sensitive, and whereas the immunoassays probably don't have the same sensitivity. So I don't know what the real answer is because no one has done that study yet. But that's a good thing to keep in mind.
Thank you, sir.
Thank you. Okay. So what are some factors that might affect your test results? And I think there are a number of things. Number one, it's pre-test variables. So how are you able to get a good representative specimen? So if you don't get a good, say, nasopharyngeal swab, you might get a false negative result. Specimen integrity. Once you see the specimen, on its way to the lab, is it held in a hot car or etc.? It really should be on an ice pack and refrigerated and taken over to the laboratory. So otherwise, the virus has a possibility of degrading. And also timing of when you take the specimen during the course of an infection. So viral shedding appears to begin 2 to 3 days before the start of viral symptoms. And then the viral loads decrease with time. So so the best detection seems to be within 1 week within the onset of symptoms. So if you were to test, let's say, 2 weeks post onset of symptoms, you may not be able to detect virus. So it's the timing is important. And lastly, anatomic source, and I'll go over that in the next few slides. And so the CDC put out this information, and I'm just copied this from the website, that for initial diagnostic testing of the virus, you should get a upper respiratory specimen. And they list the various different types of specimens here. And these different types of specimens in the upper respiratory tract have different sensitivities themselves. So in two studies that looked at the same group of patients and sampling different parts of their anatomy, they wanted to see could they detect virus. So this is how they was broken down. And so looking at nasal, and they didn't say nasal or nasopharyngeal, they said their detection ability was 63 to 73%. And the oropharynx is a little bit lower. So it for some reason it's lower. And I imagine it's just where the virus replicates, probably in the nasal pharynx. And interestingly, if you got sputum or did a bronchoalveolar lavage, your sensitivity was better, which kind of, I guess, could make sense because maybe the virus just goes down deeper into your lungs as the infection progresses. So detecting the virus also depends on where you sample.
Dr. Chan,
All right.
could you go back
yes.
to that? If you go back to that slide, I think, I've seen these before, and it's confusing to me. We do sputum for TB, of course, and that's what I do usually.
Yeah.
And so you look at this and you say, why aren't we using sputum? Why are we putting patients through the suffering of an NP swab? And because, I've heard clinicians say if they were surprised by the negative result, the first thing they'll do is go back to the patient and say, did you get swabbed? Yes. How was it? Well, it wasn't too bad. Well, then they know that it wasn't a good swab because it didn't make the patient cry or feel like they needed to vomit. So a sputum, of course, is much easier to get. However, I did see some data that showed the sputum looks better, but probably the nasopharyngeal swab gets positive at a high amount earlier, so that you can save a couple days early in the diagnosis if you use a nasopharyngeal swab, even though the sputum itself might be equal or maybe even a little better in the overall final diagnosis of the disease. Over.
Right. So it's very complicated. It's not only source, anatomic source, but it's also timing. And that's something that's not captured on this slide, but I kind of alluded to that. Yes, that's very appreciated that you mentioned that, because the nasopharyngeal swab, probably in the early parts of the detection, is good, and as you mentioned, the sputum or bronchoalveolar lavage currently persists a little longer in its ability to detect. So it's complicated, and I imagine why they used nasopharyngeal swabbing, because that's kind of like the accepted or known method of detecting viruses like flu and other viruses. So they just probably use the same methodology.
Yeah, I wish the antigen testing would just fix all of this so we could just poke a finger instead, but it sounds like there's some real sensitivity issues, and 80% sensitive in a virus that spreads like this so quickly probably isn't good enough. Over.
Right, agreed. And so we've had a couple of cases here when one patient, he was highly suspicious, or PUI, and they did a nasopharyngeal swab, negative. The doc was really persistent, and he said, let's do another. I did the nasopharyngeal swab, negative. He was really suspicious, and they did a bronchoalveolar lavage, and that was positive. So we didn't know where he was in the timing, but if your suspicion is high, I guess the take-home message is try another source. Yeah. So yeah, it's complicated. Okay, I think we're coming at the end. So when you assess these various tests, you look at the different characteristics and see which one works. There are also things to consider are availability of supplies. As you know, the world is basically suffering from a lack of reagent supply chain. It's not stable. So certain tests will have better, and maybe and it also depends on time. And other things too that, believe it or not, make a difference are just the ability of getting swabs and the media, the culture media that you use to hold the swabs in. Cost is another thing that we didn't talk about, but that's something you always consider. And then who will interpret the test result? So people in the know, they should know that that not all tests are the same. Antibodies testing is not the same as NAT. So this is kind of the information that people need to know when they interpret test results. All right, so here's my summary slide. We talked about the different types testing for SARS-CoV-2, 3 broad categories: NAT, immunoassay, and serology. We talked about how the different test principles of how NATs amplify nucleic acid, immunoassays detect viral proteins, and serology detects antibodies. And then lastly, we talked about the various different characteristics, and all these tests are not the same. So when you say molecular, you wonder what kind of molecular. So there's different different characteristics and different and all these different variables that are in play when you assess all these different tests. And I think that's it. Yeah. So my last slide is just the references. So I'll thank you for your attention, and I'll take any questions if you have them.
Thank you, Dr. Chan. That was excellent. Really, really good. And clearly you understand our capacities in the region, and you focused on the tests that we most needed to hear about. We appreciate that. Don't worry about the start. It was definitely worth it. I've interrupted you enough and asked enough questions, so let's open the floor to other questions from the region for Dr. Chen regarding labs. We know we're in the North Pacific anyway, in the enviable position of actually having labs that might expire before we even get a chance to use them. We're talking about trying to share them somehow so that they all get used. Testing persons under investigation, testing folks who are being brought in, even from COVID-free areas, who are being repatriated. So the lab testing supplies, which are a global problem, with the great work of HHS and CDC, IRR, as well as PIHOA, who's really put this on their shoulders for all of us in the region to make sure that this sticky lab issue is relatively under control for our island. So I think, you know, you folks are prepared in terms of labs, which puts us in a really good position. Are there any questions for Dr. Chen? Let me check the box. I know we're at the 2-hour mark, so I'm sure most folks are, even if they have questions, are getting ready to go see cases and get back to work.
Dr. V, this is Maria from YAP. I have a couple of questions.
Okay, Maria, go ahead.
Thank you. Thank you, Dr. Chen. This is a wonderful presentation. My first question was partially addressed by you and Dr. V, and that's regarding the source of specimen. So I understand that at the initial first few days of the infection, nasopharyngeal swab is preferred. I see from your presentation that the nasal has a sensitivity of 63 to 73.3%, which is much higher than the oropharyngeal swab. We have been collecting nasopharyngeal swab in doing influenza testing. So I presumed that would be the best source of specimen for COVID testing. Now that I see that the nasal swab is more sensitive, I'm thinking about the asymptomatic patient. Would you suggest that we do nasopharyngeal swab on them for testing? Over.
Yeah, that's a good question. I don't know if anyone really knows. I mean, you would imagine the viral loads in an asymptomatic person are more or less the same than a symptomatic person, because as you all know, asymptomatic people can spread virus. So there must be a certain threshold of virus in them to spread. So I'm just going to assume that they follow the same viral kinetics and infection as a symptomatic person. So probably in the nasopharynx as well.
All right, thank you. And that's very useful, especially for those patients who may be like small children that may be difficult to get a nasopharyngeal swab from them. So that's good to know. My second question is regarding the persistent positive COVID patient. Would you suggest that if a patient is already, they don't have the symptoms anymore, but they continue to be shown positive on PCR, would you suggest that we do serology test on them to determine if they have antibody level detected in their specimen that would be evidence enough that these people would be good to release into the community? And that would be something, if that's the case, then maybe it's good for us in the Pacific Island laboratories to make sure that we have some serology test on the side to use for that purpose. Over.
Okay, yeah, that's a complicated question, okay, because you're saying, can you use molecular testing and antibody testing to help essentially clear a person? I think right now, for the molecular side of things, so the nucleic acid side of things, we don't know with 100% certainty that if someone has a positive, let's say someone who's had an infection and then became asymptomatic, we don't know if that nucleic acid detected post sickness is truly non-viable versus viable virus, right? So no one really has proven that. It's the assumption that this virus could be non-viable that you're detecting with the NAT. So then your question is, can we then use antibody testing to see if the person has antibodies to this and essentially tell you that this person is has protection? And I would like to think that, but honestly, no one has done that study either. So the test for serology is a surrogate marker, and this is a topic that Dr. Alera addressed in this talk. That the antibodies that are being detected in a serology assay may not necessarily be the neutralizing antibody. It's presumed that if you have antibodies, you'll have immunity, but no one really has proven that. And unfortunately, not enough time and testing has been done. We're only 8 months since the virus came on the world stage. So I think we don't have a good answer. And so what I've heard is some hospitals or entities, the way they clear patients is they use purely PCR or NAT testing. And let's say if the person has no detectable virus, then they can say, well, this person then doesn't have any virus, so therefore should be clear. And they don't necessarily use serologic testing in this in their algorithm. But this, this is going to be probably dependent on different hospitals and institutions. So to summarize, I don't know if there's a clear and definitive way to clear a patient. People are using mechanisms of just NAT testing because I guess that's the safest mechanism test to clear to a patient, and because we don't know enough about the serology yet. Sorry, that's my long-winded answer.
Yeah, and I certainly respect that answer. I think it's the safest answer. I think more and more people are getting comfortable with folks who are persistently going to test positive. I know one of the studies that was done a couple of months ago from South Korea demonstrated that none of the persistent positives were able to culture any virus. That doesn't mean they're not infectious, but it does mean that it was impossible to culture actual virus from anyone after day 9. And I do think that in general, some of the criteria from CDC is changing to use time and symptomatology, and not necessarily use labs for that, because folks, and we're seeing this often even in the Pacific with quite a number of people in Guam who are persistently testing positive. I think the clinicians are still debating about whether they can be discharged. Some have been, but in general, I think there's a growing comfort with discharging these folks who are asymptomatic, who are 10 days out from their disease, and have been at least a couple of days without fever, despite not using any antipyretics. So, the more to come on that. That's a great question. Another question? We have a couple more minutes and then we'll have to close out.
I have a question, Dr. Brostrom, for Dr. Chen.
Hi.
Please go ahead.
My name is James Hofschneider. I'm a physician on Saipan. I'm wondering whether you can comment on a kind of a change in paradigm on how we look at testing, because my understanding is that in the US, for example, they still think, I think that CDC still thinks that only about 10% of those people that are infected with COVID have been picked up by current testing strategies. And the testing strategies is PCR based and it's expensive. It takes time to get the results back. But the viral kinetics is such that it's not like TB. It takes days, right? I mean, it takes weeks. This is virus that gets to you, can be spread before symptoms start and then goes up rapidly and comes down. So I heard about this idea pushed by an epidemiologist from the East Coast, Dr. Michael Mina at Harvard Public Health, which looks at a new way to perhaps test using a strip, like a paper strip, like pregnancy tests, cheap monoclonal antibody impregnated test, but less sensitive, but doing it daily. So he's talking about $1 a day type of testing. So everybody gets, the student parents can test their kids. The military can use it. Colleges can use it. So it will detect only the threshold of viral transmissibility, because even that study that came out of NYU about the Abbott test, and it says, oh, it's only 50% sensitive. When they compare to the where the Cepheid test was positive, but the Abbott was negative, it turns out that all the CT values are about 30, about 35. So those are probably subgenomic. That means just RNA fragments that are not replicative competent. They will not be infectious. So perhaps that's one way, instead of, to kind of control this virus that can transmit before we have symptoms. And it's a cheap way. And I think that's probably appropriate for us also here in the Pacific, if cheap tests, and perhaps maybe you can comment on that. Less sensitive, but fast.
That's
fast,
essentially what you're asking or saying, is that we should have a cheap and fast way of doing tests. I completely agree. If we could have something that could be tested daily, then you can kind of essentially have your finger on the pulse every day, rather than maybe once every week, or because costs are prohibitive for molecular testing. I agree. And the test that you talked about, the strip of paper, that's essentially the lateral flow assay that I described when I talked about the antigen testing. That's essentially what it is. The strip of paper is impregnated with antibodies that are specific for the viral proteins. So if we could have something like that has great sensitivity, then that's great. The chances are there's going to be lower sensitivity, but I guess that's okay if you do it every day.
Yeah, that's
the trade-off.
That's the trade-off. The point is you do it frequently, and you want what you want to do is be able to detect the virus when it's transmissible, not when it's already most of those. I think we detect virus when they're on the RNA, when it's already on the tail end of the patient's infection, right?
Yes.
A lot of those, but if we detect it as it's going up, and maybe you do it once a day, because the viral kinetics, we have to chase the virus, right? It's fast. It's not like TB. So we gotta get faster tests, and maybe less specific. Less sensitive.
Sensitive.
Even if it's but you do it daily. That's the kind of the idea. Anyway, I'm kind of excited about it. But
I agree.
Open up our community.
If you had a pack of those strips, and you just had, say, a classroom, and you just tested every kid every day, and then let's say you miss one, but probably in time you'll catch them the next day, and then you hopefully can limit the spread.
School.
Yeah.
Yes, at home before they go to school. Yeah. Anyway.
Yeah, I know that this discussion is going on every day at the CDC labs. I've seen some of the results of the early testing for these, and folks that are talking about how it could possibly be implemented. So, you know, watch this space. In some ways, James, we're asking this paper strip test to replace the vaccine, and that's a big ask for a test. It may be a race between a rapid test that can be that's inexpensive and can be used every day, or a usable vaccine that can protect us from either being infected or at least protect us from getting sick if we do get infected. I think I'm going to have to close the questions for now in respect for time and all of the folks who have logged on here. And I really appreciate the comments, and there's another great session. There are several great sessions that occurred before this. So you can go to the PIHOA website, even pihoa.com if you need to, and you can link to these lectures. We're still interested in if you have additional sessions, we can add them on. We may take a break after next week, but we can start them up easily enough as new information becomes available. You folks are on the front line. You deserve the best information that's available. We'll keep trying to find these great speakers like Dr. Lair and Dr. Chan today, who did an amazing job to digest and prepare this information for folks who are delivering care to people like you are doing. So I think with that, we'll close it. One comment is that I may update the next week's talk depending on, I know there's a recent talk that was given on COVID and diabetes, and I think that would be a priority lecture that we should share. It might be recorded. I'll keep looking. Otherwise, please send me your ideas. Watch this space, and we'll close for today. Thank you.
Bye.